Hello, my name is Arun K. Rishi. I am a professor at the Department of Oncology in Karmanos Cancer Institute in Detroit, Michigan. Today I'm going to talk to you about targeting of NF-kappa B to enhance chemotherapy efficacy in the breast cancers. The NF-kappa B pathway is a very complex pathway, and as it is well known that many DNA damage inducing therapies are used as first-line therapies for many treatment of many cancers, particularly the chemotherapy such as Cisplatin, Doxorubicin, Gemcitabine, 5-FU. They function by inducing DNA damage and are used as anti-cancer agents. However, it is also known that these DNA damage-inducing therapies activate canonical NF-kappa B pathway. The purpose of this activation of canonical NF-kappa B pathway is to activate DNA damage repair genes so that the DNA damage can be repaired. Also, the pro-inflammatory cytokines that can promote the survival of the cancer cells, as well as the immune checkpoint ligands that can contribute to the immune evasion. As a combination, then these pathways serve to promote cancer cell survival, immune evasion, drug resistance, and metastasis. The NF-kappa B pathway is actually a very complex pathway. It has a number of sub-pathways, particularly the ones that actually end up activating the heterodimer of P50 relay are categorized as atypical canonical pathways. Atypical pathways are activated by the genotoxic stress and as well as by the hypoxia or the oncogene stress signaling. The canonical pathways are activated through the receptor by TNF and other ligands. The purpose of these pathways are to actually activate an ability of P65 and relay to be able to translocate to the nucleus to activate the select NF-kappa B target genes. Another pathway, which is a non-canonical pathway that exists, that doesn't involve the NF-kappa B gamma kinase subunit, which is also named as NEMO. This pathway actually ends up activating a different heteromer, which is P52 RLB. And this P52 RLB actually activates a different set of target genes. Collectively, this NF-kappa B pathway serves to control tissue homeostasis, survival, proliferation, as well as apoptosis signaling. The DNA damage-induced NF-kappa B activation is actually activated through this circuitous pathway indicated in the box here. The atypical pathway actually is induced following the DNA damage. So on the left here, the DNA damage actually induces or activates simulation of the NEMO. Once NEMO is simulated, the simulated NEMO goes into the nucleus, and in the nucleus, the ATM-ATR kinase phosphorylates NEMO, and following simulation and phosphorylation of NEMO, the ubiquitin kinase actually then monoubiquitinates NEMO. So together, this post-translationally modified NEMO, along with ATM and other proteins in the nucleus, migrate out of the nucleus, and NEMO proceeds to participate in the assembly of the active IKK complex, which involves IKK alpha, IKK beta, as well as other co-activating proteins. The purpose of this complex is to be able to phosphorylate inhibitory protein in NF-kappa B signaling pathway and promote the free pre-P50 relay heteromer to be able to translate to the nucleus. The NEMO protein interacts with signaling transducer CARP-CCAR1. Let me give you some background of the CARP1-CCAR1 protein itself. CARP-CCAR1 protein was actually identified by us about 20 years ago. It was found as a novel apoptosis signaling protein when the cells were subjected to or exposed to DNA damaging chemotherapy such as doxorubicin and etoposide. The CARP1 is also later on found to be to regulate Notch signaling and also inhibit EGFR MAPK signaling in the worm C. elegans. This was published subsequent to our finding around the time of 2004. Later on, it was also found a proteomic study came along which demonstrated that CARP1 is a part of the NF-kappa B proteome. In this study, it was found that the CARP1 is a member of the NF-kappa B proteome that consisted of about greater than 900 proteins. However, which member of the NF-kappa B CARP1 interacted was not resolved. CARP1 was also found to regulate apoptosis signaling by EGFRs and was found to have multiple epitopes, and these were our studies, and as well as its homologs in lower eukaryotes and plants, clearly suggesting that CARP1 signaling is likely evolutionarily conserved. CARP1 is also a co-activator of steroid thyroid receptor and P53. This study demonstrated that not only that CARP1 co-activates steroid thyroid receptors to promote growth, it also co-activated P53 when the cells are treated with DNA damage-inducing therapy such as Doxorubicin. The CARP1 is also required for gene activation by Wnt beta-catenin signaling as well as encourage-independent growth of the human colon cancer cells. Together, these and other emerging studies have revealed a facet of CARP1 being a biphasic regulator of cell growth as well as apoptosis. CARP1 expression, actually, in our initial studies, we found that it was found in the perinuclear region or the cytoplasmic region. Here in the patient AML2 cells are stained with CARP1 in the cytoplasm, as well as the human bone marrow, where the megakaryocytes are stained with CARP1. In the tumor scenarios here, we have A, C, E, and G, which are the normal tissues of breast, colon, lung, and prostate origins, and the B, D, F, and H are the respective cancer tissues. What we noticed here is that in the cancer tissues, CARP expression is diminished or absent. The CARP1 signal transduction happens in many scenarios. Particularly, CARP1 regulates stress-induced apoptosis by chemotherapy, serum withdrawal, as well as EGFR inhibition. These aspects were also studied by us and others. CARP1 also translocates between cellular compartments, and as you saw that it is present in the cytoplasm, but in the subsequent studies I'm going to present, CARP1 also is present in the nucleus where it interacts with the NEMO protein. CARP is a post-translationally modified protein, which is at least modified by phosphorylation and ubiquitination. We have studied the phosphorylation aspect, and the ubiquitination studies were published by others as part of a greater mechanism by which CARP1 is post-translationally modified. Coming back to the CARP-NEMO signaling, we discovered that CARP1 appears to be required for NF-kappa B signaling by Adriamycin doxorubicin. This is a very interesting experiment which we had done some time back. And in this experiment, we utilized the breast cancer cell models where the wild-type breast cancer cells and their derivatives which express lowered or reduced CARP1, that is CLON9. In this study, we exposed these cells to the different agents which will activate NF-kappa B, such as TNF-alpha, Adriamycin, and a CFM class of compound. Here, the Adriamycin and CFM class of compounds are apoptosis inducers. TNF-alpha is actually a well-known activator of NF-kappa B signaling. What we discovered here was that in the wild-type cells, there is a significant activation of NF-kappa B in all the three agents, in the presence of all the three agents. However, when the CARP1 was reduced, we found that the activation of NF-kappa B was diminished. This was clearly surprising to us, as our hypothesis has been that CARP1 activation through the doxorubicin or CFM4 signaling promotes apoptosis. So if you were to lower the apoptosis transducer, you would expect a greater activation of NF-kappa B to promote survival. However, we noticed something totally opposite. So this meant that CARP has a distinct function in regulating NF-kappa B signaling through the DNA damage therapies or DNA damaging inducing agents, as well as TNF-alpha signaling. We proceeded to then investigate this mechanism, and to cut the long story short, we found that CARP really, in fact, interacted one-on-one with NEMO. The interacting epitopes were in CARP1 was located within amino acids 566, 580. Keep in mind CARP being a protein of about 1150 amino acids. NEMO, on the other hand, had a CARP interacting epitope that was located within amino acids 221 to 261. NEMO interacting epitope of the CARP1 again was found to be highly conserved in various species, including the fly. So in this here cartoon here, I have highlighted in the blue shaded area where the CARP epitope that interacts with NEMO resides. Clearly, this means that the CARP-NEMO interaction are likely evolutionarily conserved and serve a significant role. NEMO targeting has already been proposed and investigated. In one of the examples here, as we know that the NEMO protein functions as a scaffold to assemble the IKK complex, where the IKK beta and alpha are the kinase subunits which are assembled as an active complex. In this complex, actually, the IKK alpha and beta NBD, which are NEMO binding domains, interact with the NEMO itself at the N-terminal site of the NEMO. So in one study, actually, a peptide targeting the NBD was designed, and that peptide in principle would inhibit interaction between IKK beta NBD and NEMO, therefore disrupting the complex. And this peptide was found to reduce inner ear inflammation in the mouse model. Subsequently, many other studies were performed to show the potential of the NF-kappa NEMO targeting to inhibit NF-kappa B signaling in various scenarios. Based on that, we thought perhaps one could envision a biological model where NEMO-CARP signaling could be targeted, and whether that will also affect the functions or the ability of the cells to survive by chemotherapy or apoptosis signaling. Here we generated the breast cancer cell lines in a model where we overexpressed the CARP interacting epitope of the NEMO protein. So here we designed the breast cancer cell lines that overexpress the NEMO 221 to 261 peptide that in principle would compete for the interaction of CARP and NEMO proteins. When these cells were then treated with the doxorubicin or apoptosis-inducing CFM compound, we found that where the CARP-NEMO interaction is competed, the cell viability is diminished. So clearly it suggests that CARP-NEMO interaction likely plays a role in enhancing cells' ability to survive in the presence of DNA damage-inducing agents. This hypothesis was further clarified in the cell models where we have overexpressed CARP1 as well as a CARP1 mutant that lacks the NEMO interacting epitope. So in the wild type CARP1 expressing cells, the chemotherapy such as doxorubicin or TNF and IL and EGF class of agents nicely activate NF-kappa B as noted here by the activation of phosphorylation or P65. On the other hand, when the CARP ability to interact with NEMO is disrupted, we find that TNF, EGF, or IL beta are still able to activate P65 phosphorylation. However, doxorubicin and CFM class of compounds that activate apoptosis and DNA damage fail to activate P65 relay activation. So together these studies suggest to us very strongly that CARP-NEMO interaction has a role in regulating the DNA damage-induced P65 relay activation and consequently NF-kappa B signaling. So therefore, is the targeting of NEMO-CARP1 complex possible? Yes, we investigated this aspect further on, and we found that the two epitopes for the CARP and NEMO as peptides can be used to first study what is their dissociation kinetics here. This is an SPR-based analysis that revealed that the peptides likely bind very tight, their dissociation constant being 100 nanomolar, which is a very desirable attribute for a protein-protein based disruption analysis, meaning that one could screen for the compounds following an assay where the compounds will serve to disrupt this interaction. And in this assay, we discovered two different compounds, particularly one compound listed on the left is SNI1. SNI1 actually had a biphasic IC50 of about 300 nanomolar. And if you recall from the SPR-based analysis, the interaction between CARP and NEMO-peptides had a IC50 or KD of about 100 nanomolar. So therefore, this compound actually very closely mimics the ability of CARP-NEMO interaction. We then studied further using this compound whether this compound has any ability to modulate the DNA damage inducing chemotherapy efficacy. In this experiment here, we use the various breast cancer models. These are the PDX, murine, as well as the human breast cancer cells. Clearly, SNI itself does not impact the cell's viability, but when combined with the Adriamycin in a dose-dependent manner, we see that the cells are inhibited more than compared with their inhibition with Doxorubicin alone. So this is the one evidence that the combination of the SNI, which will disrupt NF-kappa B signaling through disruption of CARP-NEMO interaction, can enhance the chemotherapy efficacy in the in vitro models. This is in mammosphere models. We see similarly that Adriamycin in combination with SNI has a better effect on the growth of mammospheres. We further studied whether this effect is also used or is applicable to other class of chemotherapies, and here we found that indeed the SNI plus Cisplatin is also able to cause significantly more cell growth inhibition in the human as well as in murine breast cancer models. More importantly, what we tested here also is whether this strategy can also sensitize the drug-resistant breast cancer cells. Here we have the Adriamycin-resistant breast cancer models, human and murine, and clearly when we combine with Cisplatin, there is a greater efficacy of this drug combination to inhibit growth of the Adriamycin-resistant breast cancers. Now, because of the fact that we hypothesize that the activation of NF-kappa B signaling also serves to activate secretion of the pro-inflammatory cytokines, here we investigated whether the combination of SNI plus Adriamycin has a greater inhibition of the secretion of pro-inflammatory cytokines. And in here we see that in the various breast cancer cell models, whether it is wild type or drug resistant, we see the SNI plus Adriamycin combination significantly reduces the secretion of pro-inflammatory cytokines such as TNF-alpha as well as IL-8. The mechanism of this was further investigated in the Western blot analysis, and where we find that indeed, not only in the breast cancer cells, but also in the cervical cancer cells, the Adriamycin treatment does increase activation, promote activation of P65 relay, which signals the activation of NF-kappa B signaling. However, when we combine with SNI with Adriamycin, the relay activation is truncated. Of note here is that SNI itself does not cause any changes or activation of NF-kappa, the relay signaling. So therefore, SNI without the presence of the DNA damaging agent would not necessarily affect the NF-kappa B signaling at all. So this actually signals to the safety profiles of this, likely safety profiles of this compound. Further in the mechanism level, as I indicated before, that one of the important aspects of the DNA damage-induced NF-kappa B signaling is that following a NEMO import into the nucleus, the NEMO is phosphorylated by the ATM. Here we find that indeed, when we combine with the SNI, the Adriamycin-induced NEMO-phosphorylation by ATM is significantly diminished. Not only that with Adriamycin, but this also happens in the presence of Cisplatin as well. So clearly it suggests that the CARP-NEMO interaction has a wider role to regulate DNA damage-induced NF-kappa B signaling through impacting the ability of the ATM to phosphorylate NEMO itself. Whether the NEMO-CARP interaction also enhances cisplatin efficacy in vivo was tested next. These are the syngeneic breast 41 breast cancer model. These are TNBC model. And here we again find that although cisplatin is able to inhibit the tumor volume, but combination of cisplatin and SNI causes robust inhibition of the tumor volumes, as well as robust increases in the T over C of the tumor. So therefore, the tumors hardly grow very much when we treat them with combination of cisplatin and SNI. Here, the SNI is administered subcutaneous intraperitoneally, whereas cisplatin is administered intravenously. Consistent with our in vitro findings, we find that indeed in these animals that were treated with different agents, their serum levels of inflammatory cytokines are reduced when in the animals that were treated with cisplatin plus SNI. So clearly, our in vitro and in vivo data support our hypothesis that blockage of or inhibition of NF-kappa B signaling through NEMO-CARP disruption or targeting of NEMO-CARP binding can have an impact on the ability of the efficacy of the chemotherapy. So in final, then one can conclude that the DNA damage signaling, as I indicated before, causes NEMO simulation and then NEMO import, and it has also I have not shown you the data. The CARP1 is also present in the nucleus, and in the nucleus CARP and NEMO complex is then NEMO is phosphorylated by ATM, ATR, following by the NEMO further ubiquitination, and then this complex, along with other proteins which are part of this complex, migrate out to activate the IKK complex. In conclusions, then, we find that the CARP-NEMO interaction activates DNA damage-induced NF-kappa B, and we also have shown in a separate study that CARP also interacts with the RIPK, and RIPK is also part of the the NEMO RIPK CARP proteome within the nucleus. And I have not presented you the information on that, but this is a multi-protein complex that functions to activate the IKK signaling or NF-kappa B signaling following the DNA damage. SNI enhances efficacy of DNA damage-inducing therapies. So in final, then, the cartoon presentation shows that the genotoxic stress for a lower, shorter time or the lower levels of genotoxic stress does induce the NEMO translocation into the nucleus, and likely with that, the CARP and RIPK are also translocated into the nucleus. And in the nucleus, the NEMO is phosphorylated by ATM. Follow that, the ubiquitination of NEMO occurs, and then simulation and phosphorylation and ubiquitination of NEMO triggers a robust complex. That complex then migrates out of the nucleus, where in the cytosol, the NEMO protein functions to assemble the active IKK. The active IKK then activates the heteromer P65, P50, and that then translocates to the nucleus to promote the cell growth survival signaling. So in final, then, I would like to acknowledge my various colleagues, particularly Dr. Gawande, Dr. Iyer, who Dr. Gawande is a medicinal chemist, Dr. Iyer is a pharmacologist, and Dr. Lisa Polin, who has contributed to the studies in the animal in vivo work. In addition, I am also grateful to our fellows, Dr. Venkatesh, Dr. Shekhar, Dr. Muthu, who have contributed from time to time in these studies. My outside colleagues who have contributed in different aspects of the are acknowledged as well. Finally, I am grateful for our VA Merit, the Department of Veterans Affairs, to support our ongoing research, as well as many other agencies who have supported our research from time to time. Thank you for your attention.